Good morning, all of you. Today is the third day of our week-long science leadership workshop. So today we are starting with Professor Shubha Tole's talk and followed by Dr. Shalini Arya. She is talking at 1 p.m., 1 to 2 p.m. Then at 3:30 p.m. we have Dr. Yoko Shimpuku from Japanese Young Academy as well as GA. And today's final talk is going to be by Dr. Geeta Yadav from Cambridge University, and she's also a member of the INIA. So we have got 4 talks, exciting talks, so please stay tuned. So coming on the first talk, Professor Shubha Tole is a very well-known, internationally known neuroscientist. She did MS and PhD from Caltech in the United States and postdoc at University of Chicago. So you might all know that Caltech is the top 2 or 3 all the time. So it's an amazing achievement, Dr. Tole. And she has been working as professor at Tata Institute of Fundamental Research in Mumbai, India for a long time now, along with her husband. So internationally renowned for the discovery of the hippocampal organizer LHX2 gene, which has been published in Science in 2008. So this is it's a major international discovery, and I would like to emphasize that this discovery Professor Tole's team has made in the soil of India. It's not a part of her PhD or postdoc work. So it's an amazing achievement for her science, and she's a winner of the Nagar Award, Welcome Award, Infosys Prize, and many more. Of course, she's the fellow of all the Science Academies India, a member of the Academic Group International Affairs Committee of the American Society of the Cell Biology. To add on, I have already shared you this mentorship article that has been published in Nature Cell Biology, and she is a prolific writer. Let me quote one of her statements: "The multipotent student in me differentiated down a neuro-committed pathway in May 1984 thanks to the life science exhibition at St. Xavier's College, Mumbai." And to contact Professor Tole, please note down her Twitter handle. I instructed the moderators of this session to share this Twitter handle in the chat box. So you can contact her. Please don't email or use anything else. Just use the Twitter handle. And you can, of course, she'll be welcome any of your questions about this session or anything about her, the research. So it's @Shubha Tole. I say again, it's @Shubha Tole, one word without any space. So Shubha Tole is also a key scientist who pushed funding agencies to support pregnant postdocs by providing technical support during their year of the maternity leave. I think it's one of the highly, noteworthy contribution that Dr. Tole has made in this, scientific community in our nation. Professor Tole had raised awareness about sexual harassment in Indian science through her one of the well-quoted publication in Current Science, and extensively written on how Indian societal norms constrain women in STEM field. That has also been published in India Bioscience. So without wasting much time, I welcome Professor Tole, and this session is co-moderated by Dr. Mudriga. Dr. Mudriga is herself is a science leader, a young science leader from Indian Institute of Technology, Hyderabad. So over to you, Dr. Mudriga.
Thanks, Felix. I would like to thank all the audience who are here with us, and also thanks a lot, Professor Tole, for giving us opportunity to hear directly from you. I mean, we keep reading about you, from you, and there is a lot of online presence from you. But I think this is especially very, very special. We are able to hear you and feel privileged, especially sitting at home. I'm sure our audience will also enjoy hearing you and feel enriched. And like Felix said, please reach out to her at Shubha Tole on Twitter. Over to you, Professor.
Well, am I audible?
Yes, perfectly.
Perfectly.
Yeah.
Okay,
Yes.
well, thank you so very much. It's extremely, how do you say, fulfilling for me to be able to reach out to such a diverse group of young people who are simply curious about the path that various scientists have taken to explore life or to explore humankind, or to explore why we are here. In some sense, we are all part of this great quest to ask, what is it all for? What are we doing here? And we ask it in our different ways. Not all of us may be doing science, we pursue it nonetheless Somebody who paints, somebody who writes a poem or a piece of music is also exploring what it is to be human and what is the meaning of it all. This mentorship and leadership is an interesting concept. I think all of us, when we're living our lives, we just sort of do as best as we can. Okay, we make our decisions as best as we can, given the information we have at the time. We take inputs as we need, and we progress in our lives. And if it all works out, then people call you to give leadership talks. So what I'm saying is, you don't know it's leadership while you're doing it. You don't know it's mentorship while you're doing it. You're just doing the best thing you know how to do at the time. Okay, so I'm going to offer you a story of just how my path unrolled itself, as it were. And if there is anything that you find resonates with you, then I'm delighted. I'm delighted, because our lives need to mean more than just what we do for ourselves. Our science needs to mean more than just our research. It needs to be shared. It needs to be, the excitement of it needs to be communicated. And everything is for the next generation and for a better tomorrow. So if in one's own career, one is not right from the beginning thinking of the next generation, I think that's more than half the goal already lost. It's never too early to begin thinking of the next generation. It's always about passing it on. It's always about paying it forward. And I think when I look back, I think that's perhaps what I've tried to do. So I'm going to discuss a little bit of science. I'm going to showcase the hippocampal organizer story a little bit, because it became quite an interesting collaboration, and there was a angle to it that was much beyond the science. And I'm also going to sort of showcase a little bit of my journey. So let me try and share the screen now. Okay,
Yes, all is well. Yes, go ahead.
Visible? All right. So I'd like to call this talk Building the Brain and Building the Lab, because that's sort of what I was doing at the same time. Studying the building of the brain, that is my subject. My subject is developmental neuroscience. I ask how this fantastic computer in our brains, how is it assembled? How are all its circuits arranged? How do different parts of the brain know where to form? Because if the hardware does not form correctly, then the software is not going to run. So the developmental mechanisms that underlie the function of the brain is my research interest, but more on that in a little bit. My PhD and postdoc has already been summarized. So I will just introduce the brain a little bit and then discuss how I began my journey setting up my lab. For those of you who are not there yet, for those of you who are PhD students, for those of you who are postdocs, I will have something for you as the talk progresses. But let me say this. When you're a PhD student, you're not only learning science and how to do research, but you're also learning how research is encouraged and stimulated and mentored. And what you see around you that works for you and works for your colleagues, imbibe it so that you can use it yourself. What you think could have been done better, think about how you would do it better. So I'm going to tell you a story about the science and the people involved in the science. And through that, I think you can see how the interaction and the atmosphere and the almost madhouseness of my lab has produced some cool findings. So let me begin with a little bit of the science and then interdigitate with the stories. So this is a cartoon of the embryonic mouse brain. Two hemispheres, they're going to form the brain, and the entire surface of it that you can see. So here's a section view through about mid-gestation, day 10.5 of a 19-day gestation. When you cut through it, and I'm showing you one hemisphere, okay, you can see that it's essentially two balloons. And these two balloons are going to form the entire brain. And this thin wall of the balloon contains proliferating progenitors, the stem cells that are going to create all of the neurons of the cortex. So the cerebral cortex is the seat of our higher functions. And when I say higher functions, I mean sensory perception, motor control, language, learning, memory. And all these structures have to be created properly in the correct locations in the brain. You can't just have them pop up here and there. And this pattern is invariant across all mammals. This means that a little mouse and a monkey and a cow and a human, all of us process vision back here. There's a map vision, somatosensory, motor, hearing is in the temporal lobes, and then you have the learning and memory center buried deep in the brain. In the humans in the temporal lobe, and here in the mouse in yellow. So this is a young embryo section, day 10.5 of a 19.5 gestation period. This is day 12, this is day 15. And you can see how this red midline, okay, seems to be special. And this red midline is going to be the focus of the science I'm going to share with you. I'm going to show you how this tiny little red center acts like a lighthouse, a signaling center, an organizer, and it creates the tissue next to it to become this yellow hippocampus, which is the learning and memory center of the brain. Okay, so I'm going to show you an experiment along the classic Spemann-Mangold organizer experiments, where we created multiple organizers and therefore we created multiple hippocampi. So that's the science, and I will now interweave the story. I should say that I started my lab with the good fortune of a Wellcome Trust Senior International Fellowship. And I was also given substantial funds from the intramural TIFR DAE funding, for which I'm very, very appreciative. Now, the Wellcome Senior Fellowship had some key advantages which really helped me start off my lab. Because I started with these hopes and dreams of finding out how the brain is built, how different parts of it are patterned, and so on. And in order to start the lab, as you know, we need people, we need students to begin to do the experiments and so on. And when I joined TIFR in 1999, I met my first challenge. I met my first challenge because all of this business about the signaling center, which we call, by the way, the hem. Okay, think of it as a large sheet of tissue and like the hem of a skirt at the end. So all this business about the hem and cortex and so on, we didn't know it was going to be the hippocampal organizer. We just knew it was an interesting tissue that's rich in Wnt genes and BMP genes. These are morphogens, potent signaling molecules. And we wanted to study it. But at that time, we was only I. And PhD students who joined TIFR were sort of already programmed by their rich undergraduate training, which in India is very rich in biochemistry and genetics, biotechnology. So the students wanted to do different things. They thought neuroscience was a little scary, a little alien. In fact, one of the reasons I was inspired to return to India is to contribute to the neuroscience effort in India, which is still only just fledgling. So, bright students will get interested in whatever it is they're exposed to, but they were not exposed to neuroscience. So I had this problem where the best and brightest of the batch that was rotating sat in my office and said, Madam, this hem and hippocampus and cortex is very interesting and very new and very fascinating, but I don't see what scope there is in it for the future. Okay, this was a very bright person making the best possible decision that they could do at the time. Okay, so major roadblock, right? How do I begin to get students interested in this? So those of you who are students, think about why you chose to join whatever you chose to join. Was it a sense of safety seeking? I know this, I understand it, and therefore I will do it. Okay, that's a reasonable approach, but it's also a safe approach. What if something totally exciting is out there that you don't know about? You're denying yourself the opportunity of even seeing that, right? So those of us who are a little more adventurous basically widen the choices that we have before us when we finally make our choice, while it is an individual's choice. Okay, so what to do? Well, TIFR was perhaps the best place I could be in for solving this problem because it afforded enormous flexibility, okay? It afforded me the flexibility of using my Wellcome Trust Senior Fellowship to essentially create a master's by research program. Okay, now what do I mean? It's well known that Indian government funding to pay students is limited to students masters and above. Okay, you can take people as JRFs who need a master's degree, but you can't take people with bachelor's degrees. Now, the Wellcome Trust funding was flexible enough to allow me to take people with bachelor's degrees. I had to write to them a detailed justification as to how I could get the work done by potential PhD students and postdocs that I had put in my proposal by instead master's students. So I wrote a detailed justification of how the projects would be divided, how I would coordinate them and so on. And they gave me 4 master's positions. Then the fun started. With incredible support from my department, we essentially advertised in Bombay at the time a Masters by Research program. And we said, look, there are so many people interested in doing research, and maybe they want to do their PhDs later, maybe they don't. Maybe they just want to figure out what research is all about. Maybe before they decide what they want to do for their PhD, get a taste of research. So at the time, this was in 99, okay, more than 20 years ago, master's programs all over India did not have the 6-month dissertation project. So the projects that most of you have benefited from were not there. So that meant master's was only by papers, and one didn't know what research was, and one didn't have a broad, sense of what's going on in the research field in all possible areas of science in order to make one's choice. A master's program that's research heavy also allowed people to decide where they wanted to do their PhD, what they wanted to do in. So for these reasons, this program was immensely successful. And I'd like to introduce actually the first 2 members of my lab, my wonderful master's students of whom I'm extremely proud, Aditi Vyas and Sharda Bulchand. So Aditi is in industry now. She went on to do her PhD in Strasbourg and then returned and works in Nicholas Piramal. And Sharda Bulchand went to Singapore for her PhD, and now is in Singapore as a faculty member at the National University of Singapore in a very interesting sort of education and research space. So they've made wonderful careers for themselves, and I'm so delighted that they started out with me. The lab behind them was literally, pieces of wood and no shelves and no bottles. And we made all the decisions. And when the first chairs came in, we took photos, videos of them spinning around in the chairs, because until then we only had some broken stools to sit on. We've had so much fun starting this up. And this was the beginning of our Masters by Research program, which is now flourishing. Now TIFR funds it. It's no longer based on just Wellcome projects. And now it's nationwide. So students apply from literally all over the country. We get thousands of applications to pursue a 3-year master's degree, which literally gives them a good taste of what research is before students want to do what they want to later on. So in some sense, this arose out of a tough situation where nobody seemed to be interested in neuroscience to join my lab. And I simply sort of addressed the problem by thinking, hey, let's go to a younger crowd who are curious and willing to taste it for their masters, right? And this resulted in a tremendously exciting group of people in my lab. So eventually PhD students did join. So here is my first student, Bhaskar Saha, who is currently a teacher of neuroscience and life sciences at St. Xavier's College, Mumbai, my alma mater. So I'm so delighted that one of my students is there. And he is a much loved, much admired teacher. I could see that from the beginning, because all of his presentations and so on during his PhD used to be, just wonderful. This is Hari Padmanabhan, who was one of the most creative students I've had, who would like to do many things and moonlight. He decided he wanted to work not only on mouse, but on fly. So he began doing kufia experiments in the fly lab, Veronica Rodriguez, in our department. So, hey, we developed a collaboration, because I believe that students who really want to work on something and who know that they're going to invest the time and learn what they need to know should be encouraged. So we developed a collaboration, and as a result, my lab actually has a fly paper, which, people say it's very clear it's written by a mouse person for all their mouse friends to understand, a fly genetics paper. And Hari is the first author on that. He's now working with a company in Boston after doing a postdoc at Harvard. He's working with a company in Boston that uses novel CRISPR technologies and so on. So it's wonderful to see one's students, join your lab and do something interesting and move on. But so these are the 2 PhD students. What I'd like to showcase here is the wonderful master's students. Okay, that's Ryan Remedios, a really out and out creative student who could see things in sections of brain tissue that other people couldn't see. And his master's thesis included a Nature Neuroscience first author paper. He's now a faculty member at the Helmholtz Center in Germany, after doing a PhD in Germany and a postdoc in Caltech in Dave Anderson's lab. This is Vanisha Lakhina, one of the most daring students I've had. I remember she read a paper and bounced into my office saying, hey, look at this. They crossed a GFP mouse with their mutant and then made these cultures. I would like to get a GFP strain and cross it to our line and then do these cultures. And as a result, for her master's thesis, she had a first author paper that basically found something new in the field of thalamocortical development, how sensory nerves innervate the cortex. And she did her PhD at UPenn and postdoc in Princeton. Mugdha Deshpande was a student whom I shared with Veronica Rodriguez. So as a result of people like Hari and Mugdha, I actually had a whole fly angle in the lab. And this expanded me. I never dreamt I would work on flies. As a PhD student, I used to think of them as creepy, crawly things. And I regret that I never took a fly course when I was a PhD student, because then I ended up having to write a fly genetics paper. Aditi Falnikar. Aditi Falnikar did her master's with me on the same project that Manisha started up. Did her PhD in Drexel University and is a postdoc now. Aditi had a parallel interest in science communication in Marathi. Okay, fluent Marathi speaker. She in fact studied in Marathi until 10th standard, I think in Parle Tirat Vidyalaya, Ville Parle in Mumbai. And what she helped me do is that when anytime our lab published something interesting, and I would write a public outreachy little sort of blurb or press release or something, Aditi would partner with me and translate it into Marathi. And sometimes the Marathi version was actually more beautifully phrased, more metaphorical, more poetic than the English version. So Aditi has actually written about some of our lab's work in Marathi, and she's a published author in this space as well. So this is an angle that I greatly admire. Science outreach is something I'm passionate about, and my whole lab participates in science outreach. And Aditi added a vernacular language angle to that effort. It's because of students like Aditi that I began brushing up my own Marathi and Hindi. I'm Maharashtrian by birth, and I've studied Hindi in school. So now I do give, as best as I can, talks in perhaps better in Hindi than in Marathi, but I do try, because I think it's so important to reach out to the local community around you. And I encourage my students to do the same. At TIFR, we have a fantastic outreach effort beyond just my group. We have a whole team of people that is passionate about this. And I think this is one of the wonderful things of science. So this was the team that started out. Okay. And here is the kind of crazy fun we've had. On my lab page, you will find lab quotes. Don't ask me what Lakshmi without an X is, but Lakshmi was very, very particular that her name didn't have an X. Sometimes in our presentations in the department, we would announce quite happily that we will do the experiments after Hari starts breathing. And everybody would start tittering and laughing, and we wouldn't know why. Or we would announce, so we took brains from Shobna Sharma's lab, and people would be on the floor rolling. Sometimes we would be telling people, oh, I forwarded you his proposal, when we meant a research proposal, of course. Or we would tell somebody, hey, your brains are thawing. Or somebody would show up in the lab and announce, I have 4 pups, I have 4 pups. Oh, we've had fun. And each of these things shows up then on our website. So back to the science. What's the story I want to showcase? Okay, I'm going to show you a little bit of the science and interdigitate it with the challenges and the solutions we came up with. Okay, so here's the mouse brain. And these are all in situ mRNA expression detected by a non-radioactive digoxygenin procedure. So you can see LHX to mRNA is in this entire sheet of tissue that's going to be the cerebral cortex. The hippocampus will form here, but it doesn't know it yet. And this is the neocortex, cortical primordial. And this hem, which I referred to before, it's marked here by the expression of a Wnt gene, Wnt2b. You can see such a nice sharp boundary, right, between LHX2 and Wnt. So the first thing we did was we looked at Well, what happens in the LHX2 mutant? What happens if LHX2 is not there? That's one way of asking, hey, what is the gene good for? We obtained the mutant from somebody in the NIH, Denny Porter, who had made the LHX2 mutant in Heiner Westphal's lab. And we found, to our surprise, in the LHX2 mutant, there appeared to be little, if any, cerebral cortex. And in fact, what was there was a great big expanded hem. So we wondered, is it that the hem is just overproliferating and the cortex is just dying? That was a boring possibility. Or is it that in the absence of LHX2, this tissue can no more hold on to its cortical primordium identity and instead now gives up and becomes hem, which is an LHX2 negative state, self-fate state, right? So the boring possibility was already kind of discounted by the fact that Denny Porter had himself characterized the brains of these mice and had shown that there is no overproliferation in the brain, nor is there extra cell death. But we couldn't just make that conclusion, right? We couldn't just say, okay, in the absence of LHX2, all these cells become hem, without actually doing a very careful experiment using a sort of mosaic technique, a chimeric technique, where you have cells that are mutant cells that are sprinkled in a wild type cortex. So sort of classic phylogenetic techniques where you create a mosaic, small patches of null in a wild type, to ask, well, does the null patch become the hem? Okay. Now, after we published this was my lab's first paper, Bulchand et al. Sharda Bulchand's the first author, 2001. A few months later, a group from Harvard published similar findings and extended them. Okay, so we realized that there were other people on our tails. Okay, Ed Manucci, and this is was the first author, a postdoc at the time. And this is something that's going to come up later, so that's why I make a point of it now. So what did we want to do to really ask the function of LHX2? We wanted to ask what would happen if LHX2 mutant cells were sprinkled in a background of normal cells, right? This work, by then I was funded by the Swarna Jayanti Fellowship and also other grants from the DBT and my welcome trust. And this is new, so I'd like to acknowledge that. Now, to ask this kind of a question, we really needed to do something crazy, okay? What we needed to do was to make a chimera. What we needed was to embed mutant cells into a wild type, early stage embryo, okay? And then make an embryo that's salt and pepper. Now, for this, we had to get an LHX2 minus embryonic stem cell line. Okay, we had the mutant, but the mutant is embryonic lethal. So we couldn't just take its inner cell mass and make a null line. We had to maintain the mutant as a heterozygote. So we were in a quandary. And here is the giant team that by this time had joined me and were excited to do this project. So I'd like to center stage Vishakha Mangale, who joined as a postdoc with a real courage and daring to do something completely new. Nandini Gokulchandran, who was an MD doctor who wanted to learn stem cell technology. Okay, Nandini is now a co-director of a company of her own, Neurogen, and Vishakha is working in an industry, a GE Healthcare in Bangalore. Satyaki was one of the master's students, and Satyadeep Chikpire was a veterinarian, an MVSC trained person who joined us. So look at the diverse team that comes in to do this. Lakshmi was a master's and then a PhD student who had the key insight about the findings I'm going to showcase about the hippocampus. She would sing beautiful old Hindi film songs while she pipetted, which people loved. Jolly was a SRF in the lab. Ashwin was an integrated PhD student, and Ben Martinoga was a postdoc from the UK who worked with us for a short while on this project. Okay, so this entire team took this on. And I'd like to also thank, so this brings out the collaborative nature of science, right? One should just not hesitate about asking for help and collaborating. So Panikar's lab from NCBS actually trained Nandini Gokulchandran in how to take LHX2 heterozygous mice, cross them, take out the inner cell mass, and make a minus green background, GFP background LHX2 line. Shamla Mani's lab, then at the NBRC in Gurgaon, Manesar, trained Nandini in how to maintain and grow embryonic stem cells and keep them as ES cell-like. And Satish Kumar's lab at the CCMB trained Vishaka and Satyaki in how to make these aggregation chimeras. So we learned the technology there and we brought it to our lab. So it's really wonderful how a whole community came in to help us. Okay, but we didn't really know what we were doing. So this was not easy, right? 7,000 aggregations and a whole year of this later. Okay, we were not going anywhere. And Satyaki, whose master's was about to finish, began swinging from trees in desperation. And then finally we had success. Okay, so this is a chimeric brain. This is a brain in which the black cells are all normal and the green cells are mutant, LHX2 mutant cells. And excuse me, this is a control chimeric brain in which the green cells are simply wild type cells derived from the same LHX2 mating, where you get plus, 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 minus, and minus, minus. And basically, broadly, the morphology is fine. And indeed, when we do in situs, we find that a cortex marker, EMX1, and a hem marker is nicely complementary in these wild type chimeras. So this means that doing this process does not interfere with the patterning of the brain. Now for the mutant, okay, in the mutant, look how the green patches seem to all overlay with this hem marker LMX1A. In fact, these are the same section, first photographed in green and then done the in situ. So I'm going to overlay them for you. Look, if you will, at this triangle, or look, if you will, at this thin line. Okay, the green cells are mutant and they are flanked by black wild type cells. So if there was any non-autonomous rescue to be happening, you would see it here, but no. Any cell that doesn't have LHX2 can't become cortex and becomes hem. Okay, so this was extremely cool because this means LHX2 cell autonomously selects for cortical cell fate. LHX2 is a cortical selector, and we were tremendously excited about this. I went to a neuroscience meeting in the US, the Society for Neuroscience, the largest gathering of neuroscientists all over the world, at Atlanta in 2006. And then I found that Ed Bonucci's lab had a poster using a completely different approach to get similar findings. LHX2, in his hands, they'd made an LHX2 conditional knockout, and using a Cree, tamoxifen-driven Cree approach, they'd found that LHX2 null cells also become hem. Okay, so this could have been terrifying and horrible, and we'd be worrying about scooping each other. But I think this was two scientists that didn't believe in this sort of cutthroat, kill or be killed kind of approach to science. We actually had a 2-hour conversation sitting by our posters. And we said, look, our people have worked really hard on this. Okay, and each of our stories, while exciting, is not complete. So rather than go back home and wonder how far have they gotten, let's submit a half complete story and just to get ahead and scoop the other, why don't we just keep each other posted? Let's just be completely frank and open and tell each other where the story is at. Whoever manages to finish their story properly first, at least the other person knows that that's where it is. Maybe the other person can also wrap up their story and submit it at the same time. And this was an incredibly trusting collegial equation. So we basically turned a competitive, potentially competitive situation into a collaborative collegial one. And each lab had a big file of emails and things exchanged from where we looked at what the other was doing. And it turned out then that we were each ready to sort of submit about the same time. So we decided to co-submit to Science. The reviews came back saying your stories are very parallel. So they had some common findings like this one, the cortical selector, and they had some unique findings. I'm going to tell you about the unique finding from my lab in the next few slides. But by then we had intellectually input so much in each other's papers that we were already co-authors on each other's papers. So the editor suggested, why don't you combine them? Now, again, this was an unusual suggestion, but we decided we wanted to tell the best possible story. Okay, we didn't want to just say, oh, this is my turf and that's your turf. So we combined the stories, okay? And in so doing, each lab added its unique angle that the other didn't have, in addition to this common angle. I'm going to tell you about our lab's unique angle, and that pertains to the insight Lakshmi had about the hippocampal organizer. So here, you see, we created an embryo with extra hems embedded in normal wild-type tissue. It presented us the opportunity of asking a question we could not ask in the standard null mutant, right? Because in the standard null mutant, we had an expanded hem, but there's nobody for that expanded hem to respond to. Here, in this chimeric situation, we have hem and wild type tissue, hem and wild type tissue. So we could ask now, what does an ectopic hem do? This is sort of like the Spemann-Mangold experiment where they took out the organizer and put an extra organizer into a developing gastrulating embryo to ask, what does an extra organizer do? Okay. As a result, we could publish our findings in Science, but that's a little ahead of the game. So here's the cartoon again, which you know. And here is a sub-cartoon of just the middle part of this, okay? Now, the green is the hem in the normal situation, and this is the responding tissue. Here are extra hems in the chimera. In the normal animal, the hippocampus, marked by these pink and blue fields, grows next to the hem. I will show you that in the chimera, next to each hem develops these pink and blue hippocampal fields, showing that it's not the hem that becomes the extra hippocampus, but that next to the hem induces an extra hippocampus. So here are control sections at a little older stage. Here is the hem, which by embryonic day 15 has now shriveled away, and this baby hippocampus has begun to develop. And in that baby hippocampus, here is PROX1 showing where the dentate cells are streaming. And here is KA1, it's a CA field marker. Okay, I've only got these markers on this slide right now for lack of time, but the paper has others. And here is the chimera with mutant cells, which have each become hem because they lack LHX2, they couldn't become cortex, they became hem. And next to each hem develops a PROX1 dentate patch. You can even see some streaming going on. And next to each PROX1 patch develops a KA1 patch, and so on. So essentially, we created a multiple hippocampus embryo, right? So to summarize, I've shown you that removing LHX2 makes hem. Each hem can induce a hippocampus. And I've shown you a story that was published in Science without a single reviewer's experiment to address the comments, because two groups had independently pretty much verified the central finding, and we had exciting additional findings. Now, how did we decide the authorship? Okay, I'm sharing this story because it's unusual, and this is, I think, a way that scientists could work together. Ed Mnuchin and I both realized that our people had worked enough, and we didn't want anybody to not get the credit. So this paper actually has 4 first authors on it, okay, 3 from my lab, Vishakha Mangale, Nandini Gopalchandran, and Satyaki Prasad, and the first author from Ed's lab, Carla Hirokawa. But then the key position argument about who gets first position, right? Now, in biology, the first authorship, the first location, even with this multiple asterisks paper, is important for perceptual reasons. But the senior author position is also important. Okay, especially in the US, people look at who's physically last, and that's it. Now, Ed was coming up for tenure. I had the decision to make of publishing in Science for the first time from my lab, and perhaps one of the few papers in Science from India. I wanted the citation to carry an Indian name. So I proposed to Ed, how about my lab have the first position and your lab have the last position? And of course we'll have asterisks. So this just went so smoothly when we celebrated later and said, you know, it was so simple. It was just so simple. We would have never imagined that this would happen without an argument. Of course, there were consequences, right? Of course, people thought that Ed's lab had led the show until I started giving talks about it and talking about the hippocampal organizer and talking about how it was a fundamental discovery. So eventually it all worked out. And that's why we now have the paper Mangale et al. With an Indian citation, which was important to me at the time. So, you know, to summarize this, we all did the best we thought we could do at the time. And this is how it worked out. Okay, we could have gone in so many different directions. We could have had this competitive fighting, scooping each other relationship. We could have argued forever about things, but this allowed us to basically have a collegially published discovery. And the discovery is not shortchanged by how we collaborated. The discovery is in fact enriched. So that's this angle to the story that I'd like to offer you. Okay, so what came next? Yeah, well, we certainly immediately went out and celebrated. So this is Marine Drive in Mumbai, and it's largely the same team. You can see Vishakha and Lakshmi and Nandini here. And I would say, for those of you who have just started out your labs or are thinking of starting labs, that these celebrations are in fact what people remember. Okay, these celebrations are what people in the lab will tell you that, hey, you know, we used to have so much fun. The lab was a collegial place because for people like meeting for things that are more than just the experiments and you know, science and the actual planning of the experiments and so on. People like getting to know each other under sort of circumstances where they can unwind. People have told me that the parties I've had have done so much for the lab to come together and for people to work together more happily, right? So in some sense, you don't remember all the gels you ran, but you do remember all the crazy things you did at parties. So here is a collation of that. Okay, these are collations of parties from some time ago. This child is now 15 years old. This was a postdoc we had at the time. This child is now 18 years old. And you can see we have, this is me doing our lab's dance of the hippocampus, which I will not demonstrate on YouTube, but we have the dance of the hippocampus and the dance of the neural tube and so on. And I use those when I teach my courses. So this is one thing I'd like to say, you know, leadership doesn't just mean sort of, you know, pursuing science and having people do science with you. Leadership also means bringing people together and having collegial interactions of all kinds. Okay, so I'm going to fast forward to the final angle to my story that talks about postdocs. I have been incredibly fortunate sort of after this phase. I'm going to talk about postdocs, and I'm going to also post in the chat box. I'll just exit this presentation and I will post these links in the chat box for you. Okay, because I think one of the most challenging things for students, especially students who train in India, is how to choose a postdoc. We choose postdocs just based on people's papers without actually thinking how that postdoc and the postdoc mentor is going to affect our careers and our abilities to do our research and give us our launching pad. Right? So there is much that has been written, some of it by me, some of it by people I really admire and respect. Ben Barris, now no more, Ron Dale. So I'm going to post these in the chat box, but basically just think, if you just Google the idea of planning your postdoc, okay, if you Google that phrase, you know, you will get many, many more links, because your careers beyond your postdoc don't just happen, okay? They are carefully planned. You know, it may be hard to think during your PhD of what you do beyond your next experiment, but it's very crucial to the long-term success to set a goal and then plan to reach it. So your postdoc needs to enable whatever it is you want to do with your life afterwards. If it's teaching, that's fantastic. If it's industry, that's fantastic. If it's having a research lab, that's fantastic. If it's stepping out of science and, hey, taking a law degree, that's fantastic. Your postdoc should be something that enables that future. Okay. So let me just right now exit out of this, and I will just find the chat box so I can post this for anybody who wants it. Okay. How much of this has come? Do's and don'ts, life as a postdoc, choosing an institution, choosing a lab, how, when, whom. Yeah. Okay. All right, good. Let's go to screen share again. I'm almost done, so there'll be time for questions. Okay. Okay, so I'm going to now introduce 3 people who postdoced in my lab and have moved on to their careers. People whom I'm so glad joined me and I could work with them for a while. Bhavna Muralidharan, she postdoced with me, led my lab, almost kicking and screaming, into a new molecular frontier. She did the first ChIP-seqs in my lab together with some wonderful master's students. Amongst them Upasana Maheshwari, who started out the process with her. Bhavna is now a new faculty member, an assistant professor at INSTEM Bangalore. She started her lab in something new and different. And not only was she a Wellcome Trust Wellcome Trust DBT India Alliance Early Career Fellow when she was with me, but she's now an Intermediate Career Fellow in her own independent lab in Bangalore, in INSTEM. Geeta Godbole did some interesting work looking at the interaction of LHX2 with two other fundamental transcription factors. She published really well and she wanted to work in industry, and she's got a job she really loves with Cipla. Mallika Chatterjee did work with me that is as yet unpublished and completely interesting, and has taken us into the space of choroid plexus and cerebrospinal fluid. Okay, Mallika wanted a job near Delhi, and she's got what she wanted. She's an assistant professor at Amity University. And I'm delighted that these three very talented individuals, all of whom were Wellcome DBT Early Career Fellows, have progressed in their careers to something they wanted to do. Okay, I'd also like to say that Bhavna and Geeta had children while they were doing their postdoc. And this is an important life milestone, right? You should have children when you want to have children, if you want to have children. And yet, what I've realized in talking to people, and this is not just in India, this is when I travel abroad. When I do a women in science kind of angle in my talk, I have people approach me afterwards telling me they're so worried how to tell their advisor that they're pregnant or they want to have a child, and they won't be able to work as hard, and they somehow feel inadequate as a scientist just because they've chosen to pursue their personal life goals and have a baby. I mean, how cruel is that? Okay, how cruel is that? How can we as a society make people feel bad for wanting to do what they want to in their personal lives, right? This is when my own experience, since I chose to have my children after I started my lab. So I had my children at 35 and 38, incredibly aged and old by Indian standards. But hey, you take input from many people, finally you make your own decision. That worked for me. And I found it worked for me because I didn't have to be at the bench the whole time, right? So I began thinking, how can I help these postdocs who do have to get lab work done and who do have to move their projects along? That's when I came up with the idea of basically hiring a GRF to support each one in their time. So I hired a GRF, and the idea is that person would learn all of the techniques. So then if the postdoc didn't feel quite well enough to start work early in the morning, they could phone in and the experiment would start. So basically this kind of tag team really worked, where there was somebody to move the experiment along, and the postdoc didn't have to feel guilty about, oh, things are not moving. Of course, I was extremely encouraging and supportive about flexible schedules, because to my mind, people get whatever they want to done when they are passionate and motivated about it. If you don't question when somebody is going to come into work and do it, they will find a way of their own accord. I mean, Bhavna used to, when her baby slept and her mom was with her to take care of it, Bhavna would go to the confocal at night where she had all the slots open for her, and she would image away into the night. Basically, if you let motivated people plan their own lives, the best science will come out of it. And the best thing one can do is support this. So at some point, I was part of a committee led by Shahid Jamil to discuss many, many issues. And one of the things we suggested was that we should allow institutions we should encourage institutions and funding agencies to support postdocs during this period of maternity and even after. So my postdocs had JRFs working with them for a good 2+ years to support them in even the early years of raising their children. So these are the kinds of things I think we can do. If we find something works for us, we can find a way to spread it and help others, right? So I've written up some of my story in this blog, Scientist and Mommy. I won't put it in the chat box because I'll have to exit the presentation again, but if you just Google my name and Scientist and Mommy, you'll find it. It includes the story I've told you interdigitated with how I was juggling being a mother and being a scientist at the same time. And it's just my story. There are many, many solutions to this set of challenges, as there are solutions to everything in life. If one basically figures out how to do what is good for us, each one has a solution that's good for you. Okay, so if you listen to many and then choose your own path, you know, something from here and something from there may help you. Okay, I'd like to close with a set of thoughts I had after I visited an institution in the US where I was moved by the stories of a few women who spoke with me privately about the hard time they've had as women in science, having children, dealing with issues such as, you know, elder care, having to deal with ailing parents or nursing children, and just how insensitive the environment around them was at different stages in their life. I was moved enough that this came out in the form of a poem that I'm going to share with you. I wrote this up on the plane at, you know, 10,000 feet. So it is what it is. I'd like to close with this poem before I take questions. It's my thoughts on mentoring young trainees. A young mind knocks on the door. I like your work. Will you train me? What a privilege and a responsibility. A mind that will explore and experiment, enthused, energized, immersed, needing only mentorship and direction, but not too much. But who knows what an unfettered mind may discover? My lab then, a neural network, each mind leading us to a different unknown. Young minds also need nourishment and support, intellectual, material, and sometimes practical, but they do not leave their lives at the door. A student juggling a project with an ailing parent or a personal crisis, or a postdoc brushing between experiments to express milk or to take her baby to the doctor, all deserve respect and consideration. For we are all minds linked in humankind's timeless quest to understand who we are. Young minds knocking on our doors, burning with desire to carry the torch further, asking only for fuel and flame. What great responsibility and what exceptional privilege.
Thank you, Professor, for creating that aura. I mean, I think it has created an aura that I think should not fade away around us. You've created an exceptional kind of feeling. But I particularly want to say one thing about you. You presented an example of a complete person, a researcher, a human being, how you integrated people, acknowledging people, because I know, being a young researcher myself, we are nothing without people who work with us rather than for us. So it's with us. And this is something that I think is a great message you have given to the young leaders, young scientists. At the same time, the thing that you started with, thinking about the posterity, thinking about the future generation. It's indeed never too late to start thinking about future. Thank you, ma'am, for that message. Really, really thank you a lot. I think I can keep on saying a lot of things with all the emotions that I have at the moment, but I think I'll go to the questions that people have posted on the YouTube chat box. One of the questions is by Mr. Amritya Pradhan. So he's asking, is imposter syndrome faced by most of the researchers important for their success in a way that it keeps them grounded, or is it the other way around? Over to you, ma'am.
I couldn't hear properly. Is this about imposter syndrome?
Yes, ma'am. Imposter syndrome. Yes.
This is
Is
the
it
one
important
question I
to
don't
be-
have a solution for. All of us, and perhaps more women than men, I'm told, have strong cases of imposter syndrome. Sound very confident and very comfortable and so on, but you won't believe how anxious I was about this talk, because I was invited to speak on leadership, and and when I was told there were so many people signed up, I actually began worrying, abhi in sab logon ko main kya bolu? Kyunki I'm a neuroscientist, there will be people from all kinds of walks of life. What exactly do I have to offer them? Even for this talk, I questioned whether I'm the right person for the job. Even for this talk, I thought I might be an imposter. So I have it too. When I'm on committees, I wonder, do I actually have anything useful to say? So I'll tell you, I'm possibly the only Indian scientist to be on the board of reviewing editors for science. This is not the board that actually reviews the paper, but we are a team of people to whom a manuscript gets sent for a 2-day opinion on, do you think it should be reviewed? Okay, this is a tremendous responsibility. You won't believe how many emails I have sent to the editor who sends papers to me saying, are my comments actually useful? Because, I'm not an expert in this or that. And, the editor is like, please, you're an expert in the field. Whatever you think, your gut feeling is important. That's exactly what we want. And I'm thinking, my God, but why? Okay. you understand. So we all suffer it. I don't have a solution. So I'm sorry. Your first question, I don't have an answer.
No problem. But how do you overcome that? Because I think that will be useful if you can just shed a few sentences on that.
I think the best I can offer is speaking to others who have it too. So the reason I revealed how much I feel like an imposter in so many fora is, maybe if you connect, hey, maybe everybody feels it, and the best we can do is the best we can do. If we recognize all humans are limited, finally we can only do a little. So if we recognize that our limitations are everybody else's limitations and we offer them anyways, if you have a little courage to offer it anyways, then somebody will meet you and say, that was really useful. And then you say, okay, what I had was useful to somebody.
Yes, over to you, Felix.
Yeah, what a speech is that, Professor Tole. It was fantastic. Lots of creative spark into it. Is really surprising, and it's absolutely amazing job that you're doing. Congratulations to you. And yeah, without wasting much time, we have lots of interesting questions. So let me pick one question. Vrinda from the United States. She's asking, I'm inspired to hear your story, ma'am. I'm currently a postdoc at University of California, Berkeley. After a first postdoc at Max Planck Institute in Germany and PhD from IISc, I really wanted to institute. I couldn't get anywhere. Rampant politics, so as nepotism,
I'm sorry,
reservation
excuse
policies
me.
are not conducive.
I can't hear you
sure
fully. Sorry, I can't hear you fully. Why don't you post the chat box? I can just read through them.
Perfect.
You could just cut paste, many of them, and I can pick some to answer.
Yes. Perhaps you can read out the question as well.
Okay. Broadly, this person has had trouble despite trying in many premier institutes because there's rampant politics and nepotism and the atmosphere is not conducive. She's sure there are thousands of people who want to serve India, but it looks like India doesn't want us. I can't speak for all of India. I can, however, express my deep sorrow that many institutions do not treat applicants with the basic respect they deserve. People don't reply to emails. People don't have the courtesy of saying they're not interested in the application, or even a hint as to why, saying, look, we're looking elsewhere. People don't have this professionalism, right? Now, how did this arise? Okay, human nature does not change. Okay, so the people who are running institutions today were students some time back, and we did not have such sessions for them. A student who is raised in an unpleasant environment is going to an unprofessional environment, and who is not asked to reply to emails professionally on time is not going to do it when they are deans and directors of institutions or senior admin people. So I think the way to start is changing ourselves. Okay, I can't solve the problem today of why institutions in India don't respond to emails, or why the processes seem to be unprofessional in one or the other place. But I can say we can fix it by, so it so happens I'm also coordinator of the hostel in TIFR. And one of the changes I implemented was in telling all of the people who are managing the hostel that students are not the lowest rank who can just be treated this way and that. They are the future chairs and deans and directors of institutions. And if you treat students well, and if you make them feel that the institute really cares about them, Indian science is going to improve later. my own experience at Caltech, I was treated so well, even though I was a little silly. I mean, the kinds of things I used to ask, young people will ask random things, right? And, try and say, oh, well, can I just stay 2-3 weeks more, even though the next gen is coming in. We'll ask all kinds of questions from our own little space. I was treated so well and so kindly. Okay, so if that is the change, if you treat young people like they're going to be the future leaders and directors and deans, then you're going to have excellent directors and deans down the road. So that's my broad solution. I'm trying my bit from my end. I unfortunately cannot speak for all of India, but that's what workshops like this are about. I mean, your institution has started this workshop. So that already says that you're trying to change the system from your end. So maybe we can all work from different ends and yeah.
Yes, ma'am. I'll do the next question if that is okay. I've also posted that in the chat box, but I'll just read it out. The question is from Priyanka Sharma. What's the scope of STEM cell technology in India? And I just want to expand the context a little bit.
So if you don't mind, can you post several questions so I can choose from them?
Is that okay? I'll do that.
It's a scientific question, right? I mean, what I have to say is as good as what anybody else has to say. Stem cells are rampant everywhere.
Yeah.
Reading, search, Google, find your answers. So if you can few, then I can select ones where I can actually say something useful.
We will do that. We'll do that. Just posting that to you here. Yes, done.
Okay, just give me a minute to read through them. How much time do we have? It's already 12.
Yeah, ma'am, we can do another 10 minutes, Felix.
Okay. So let me just read a few. Shriti Vishwakarma asks, can lower order and higher order brain functions be developed or enhanced over time? All brain functions can be enhanced over time with practice. It's a use it and lose it kind of approach. So the more you do something, the better you get at it. Take any motor skill as an example. Okay, if you're going to play a musical instrument, the more you do it, the better you get at it. The earlier you start, the farther you get. All circuits improve by repeating, even giving talks, which is why one tells one's students to always practice the talk before you give it. Garima Mishra asks, can I say something about what's happening in dreams? So many of these questions are best answered on Google, where there are experts better than me. Okay, so rather than give you a popular science answer, I will direct some of you to Google to look at, Google your questions, what happens in dreams, right? I mean, we clearly construct things that are bits and pieces of what we already experienced, mix them up, and we construct things that never happened either. So that's sort of the frontier of neuroscience. Vinny Lathar, what kind of change do you see in students' perception, knowledge, patience, and career orientation over the last few decades? Okay, that's interesting. I think students, a couple of decades ago, perhaps were more safety seeking. Okay, it was a different India. The job prospects were different 30, 40 years ago, even more so. Now I think students are willing to dare a little bit more. Technology and the net and the smartphone has enabled access to different viewpoints. I also think students are a little more flighty and short attention span because of this random information that comes in. I mean, you also have to develop a filter of what you read. I think that because we are now in an age where there is so much out there to read, we pay less attention to depth than we could. Who is to say if it's for the better or the worse, right? People who want to read something in depth will, despite all of their inputs, and technology is wonderful. This whole Zoom webinar would not happen without it. So we take the pros and the cons and we do the best we can.
Yeah, here is one interesting question from Anil. I think if you allow me, I can read it out, the question. Your story about Ed Monuki's correspondence and the decision to collaborate with him on your landmark paper in Science, let me relate it with the Darwin invited Alfred Russel Wallace to co-speak at the Royal Society of London, and eventually the Origin of Species, where both shared the authorship. Such a rare instance of scientific diplomacy and professional ethics is getting endangered in these times of mad rush to get headline-grabbing stories, even through preprints. How to increase such professional diplomacy among early career scientists here in India? Any advice? Yes.
So how to encourage is simply by doing it and sharing it when you do it, right? I will say we are who we are, and there is no delaying doing the right thing. If you have the attitude, oh, right now it's for my PhD, or right now it's the first paper from my lab, I'll do the cutthroaty thing there. And then later on, when I'm established, then I'll be, fair and collegial. There is no doing that. You have to do it as early as possible from the first step. Every step you take has to be from that framework with the long-term goal inside. What kind of a scientist are you? What kind of a person are you? There is no postponing good things for later. So, take risks. If you believe in something, take risks and pay it forward. If somebody has ever been fair and generous to you, pay it forward. Do it for 10 others.
Okay. So we have one more question here. Interesting. Sudhanshu from Ranchi is asking, as you are someone who worked with hippocampus and amygdala for a long time, do you believe there is a rational emotional dichotomy of the brain? As in, rational thoughts are brewed in cerebrum while emotional thoughts are in amygdala, evolutionarily primitive region. Another random thought is, I heard women are more emotional than men in general. Would that mean amygdala is more active in women than in men?
Okay, first, it's all connected. Okay, the amygdala connects to the cortex connects to the amygdala, the hippocampus is involved there. So I'm not going to deal with the first part as to dichotomy, this in one side, that in one side. That's all from this popular science stuff where people want simple stories, left brain, right brain business. People want it all simple. It's not simple. I'll deal with the second part. I heard women are more emotional than men in general. Would that mean amygdala is more active in women than in men? Thank you for asking. Courage to ask it. Okay, these days, if you call a woman emotional, you'll get beaten up. I would say it's not fair and not reasonable to call anybody anything. It may be the case, or rather, I believe it is the case, that women or some subset of people are just more comfortable expressing their emotions. Okay, show me a guy who has not experienced deep emotion, who doesn't get extremely. We just socialize men differently than women. If you work in cultures where it's not such a bad thing for men to express their emotion, you will see men who are as emotional, and it's not a bad thing. I have seen, so I sabbaticaled in Geneva for 6 months. I worked there with a postdoctoral colleague, a guy whose eyes would moisten and, would literally cry when there was something emotional going on. And, sometimes when he was talking about himself. And it is so comfortable for that to happen. We make boys who cry feel bad about themselves. I mean, that's such a horrible thing to do. And we dismiss, women who are upset about something as, oh, you're being emotional, or worse, right? So I think the words calm down are actually the biggest weapon that is used to silence somebody who's trying to make a point, because you take the argument into a different space, right? You immediately take the argument from whatever is being discussed to the fact that they are being emotional. So thank you for asking the question. Asha asks, while we try and encourage interdisciplinarity, the job prospects, especially in academia, are difficult. Okay, job prospects for an interdisciplinary trained person are incredibly good, actually. There is a lot of funding. The Human Frontiers Science Program, HFSP, I serve on their postdoc proposal evaluation committee. They look for people who are going from one field to the next. They actually look for it. And to have two entire disciplines in the two parts of your own brain as it were is actually an excellent thing. So if any institution doesn't appreciate it, don't take a job there. They are still old, stodgy in the dark ages. I mean, at TIFR, we have people trained in physics working in the biology department and the chemistry department, and so on.
Yeah, I think, ma'am, that's a good thought. I mean, if people are still stuck in the past, I think we deserve to be at a better place, isn't it? That is what it is. So I think on behalf of everyone, I really, really extend my sincere thanks and gratitude to you, ma'am, for creating, I would use the same word, aura. I think It's a very fragile thing, but it is important to come back to the same thought again and again to make it imbibed in our attitude. With that message, I think thanks a lot, ma'am. And we look forward to follow you and look at you as a good role model. Thank you very much, ma'am. Over to you, Felix.
Yeah, thank you so much, ma'am. It is really wonderful to have you here with us today. And you have actually divulged so many intricacies of the being mentor and coming back home in India and establishing a new lab with full of creative spark. It's amazing. And especially on the gender point of view, you have discussed so many interesting facets that is rampant in academia. And of course, I'm going to share all of your links to the participants, so they will also follow you through the Twitter. And more than that, I will also share the links to your blog, The Mommy and Me. So it's very interesting. I haven't. Yes, exactly. I will also read that. I'm sure there will be very interesting points there. Yeah. Thank you so much, ma'am, for being part of us today. And I would also like to share with all the audience that ma'am spoke about Welcome Trust India Alliance. Of course, the DBT Welcome Trust is the one senior career program is the one which brought her back home in TIFR. And the CEO, Professor Shahid Jamil, has agreed to volunteer his time to interact with us, and it will be on 26th June. So please stay tuned. That will be on the Friday. And also another connection, madam is also actively contributing. Her blog itself is hosted in India Bioscience. So we also have the director of India Bioscience, that is Dr. Smita Jain. She will also be on the same day, Friday. So
Wonderful.
Stay tuned. Yes.
Is Professor Shobna Sharma going to speak next?
Yes, tomorrow. Yes.
Tomorrow.
She's also here. Would you like?
Yeah.
Would you like
no.
To make
so
any...
I would like to actually say that Professor Shobna is one of the women scientists senior to me, who was a mentor and an amazing support to me when I was a young in TIFR. Yes. And she's one of the role models for. She started her lab in a very different India, much before me, and she's a wonderful example of how you can make it easier for the next gen and the next gen. So I'm sure you will introduce her more fully, but I would like to say long after her own children were grown and gone, she really nucleated and spearheaded TIFR's first childcare center. When we were required to have one back in '99, I mean, she really led it with so much of her time went to it. And, she did it for everybody else. Her children were long since grown. Right. There was a time when she was chair of our campus committee. She was chair of the department. She was chair of the women's cell. She was also the first chair of the TIFR women's cell. So, I mean, I have, nothing but admiration. And it's such a wonderful forum to say thank you, Shobna.
Yeah, we will be waiting forward to your exciting talk, Professor Shobna. It's exactly tomorrow at 11 o'clock. And it's definitely going to be another inspiring session just like today. Thank you so much, Professor Tole, for your time with us.
Felix, I just wanted to say thanks to Shubha. I've been listening to her talk, which is wonderful as usual. And tomorrow I'll refer to some of her contributions as well, in addition to what she said. Thank you.
Perfect. Thank you so much. Allow me to share my screen. Yes. So our next speech will be by Dr. Shalini Arya, and she's coming under the Young Science Leader program at 1 o'clock. So I'll see you all at exactly 1 o'clock. So enjoy the rest of the day and have a nice, delicious meal. Goodbye and see you all at 1 o'clock today.